Assalamu alaikum. My name is Sufyan Ahmad Hashmi. Today the topic of my discussion is mineralization. What is mineralization? The deposition of mineral salts in and around the organic matrix to make it a calcified structure. Although tissue fluid is supersaturated with calcium and phosphate ions, spontaneous precipitation of calcium phosphate does not take place because inhibitory macromolecules inhibit the crystal formation. Unstable initial cluster of ions needed to form a later structure. Furthermore, the formation of clusters of ions requires the expenditure of energy, and the energy barrier must be overcome for the crystallization. Organic matrix. Organic matrix is composed of collagenous protein and non-collagenous protein. Collagenous protein consists of type 1 collagen. It acts as scaffold that accumulate the mineral in holes and pores of a fibrils. Non-collagenous protein consists of proteoglycans, phospholipids, phosphoprotein, osteopontin, osteonectin, phosphopyrin. Non-collagenous protein are involved in mineralization of enamel, whereas in other hard tissue, collagen play an important role. Alkaline phosphatase, pyrophosphatase, calcium ATPase, metalloproteinase, proteoglycans, and nonic phospholipid bind calcium and phosphate ions, which later form calcium inorganic phosphate phospholipid complex. This will initiate the mineralization. After that, crystal grows rapidly and rupture from the vesicle. These vesicles then fuse with adjacent clusters and form mineralized matrix. This picture shows the collagen fiber and hydroxyapatite containing the calcium and the phosphate. Theories of the mineralization. There are three theories of mineralization. Robinson's phosphate, which is called as alkaline phosphatase theory. Second one is nucleation theory or seeding theory. Third is matrix vesicle theory. So let's start with the Robinson phosphate or alkaline phosphatase theory. Alkaline phosphatase is the enzyme which participates in the process of calcification. It resides in the matrix vesicle. This enzyme hydrolyzes the organic phosphate containing substrate and increases the local inorganic phosphate concentration. This enzyme hydrolyzes inhibitor of hydroxyapatite and provide phosphate ions for the formation of hydroxyapatite crystals. Second one is the nucleation theory. This theory was proposed by Newman and Newman in 1953. According to this theory, a nucleus is formed probably in relation to collagen, effective in aggregating calcium and phosphate ion. Then hydroxyapatite crystals grow spontaneously. Nucleation site and the nucleating agents: ground substance, sulfated glycoaminoglycans, proteoglycans, collagen fibrils, phosphoprotein including the osteonectin. Mitochondria, the early storage site of the calcium and phosphate in the form of amorphous CaPO4. This stored mineral is made available extracellularly, which causes the growth of the crystal, forming extracellularly in association with the matrix vesicle. There are two types of nucleation: homogeneous and heterogeneous. Homogeneous nucleation, any local increase in concentration of inorganic ion permits a sufficient number of ionic clusters in crystal to form. Heterogeneous nucleation, the presence of nucleating substance allows the crystal formation to occur in the absence of a locally increased ionic concentration. There are certain objections to the nucleation theory. This theory failed to explain the mineralization in enamel and cartilage. This theory also failed to explain the mineralization in soft tissue, though it contains collagens, because collagen in the soft tissue are densely packed with impede phosphate ion access to nucleation sites. Now we talk about the matrix vesicle theory. The mineralization requires the presence of extracellular matrix vesicle. Matrix vesicle are small membrane boundary structure having the diameter of 25 to 250 nanometer. They are lying free in the matrix where the calcification occurs. The vesicles are rich in phospholipid, especially phosphatidylserine, a lipid with high affinity for calcium ion. The vesicle also contains annexins, which forms calcium channel, thus incorporating the ion within the matrix vesicle. Now we talk about enamel mineralization. Matrix vesicle and collagen are absent in the enamel. Enamel mineralization is thought to be achieved by the crystal growth from already mineralized dentin. That is, mantle dentin with subsequent shape and size of the crystal is determined by the enamel protein of the matrix. Enamel portion are of two types. Number 1, 90% emelogenins, proline, histidine, glutamine, which help to maintain space between the crystal. Second one is 10% non-amelogenins, which is tephylline, enameline, amylene, which helps in nucleation and growth of the crystal. Now we talk about the crystal growth. On the left side, the picture clearly shows 1 micrometer size hydroxyapatite crystal. Hydroxyapatite is a major component of tooth enamel and bone mineral, a relatively rare form of apatite in which most of the OH group are absent and containing many carbonate and acid phosphate substitution is a large component of the bone material. Initial growth of apatite is rapid, then slow down due to the following factor. Non-collagenous protein can bind selectively to different surfaces of crystal, preventing further growth and finalize the size. Accumulation of inorganic pyrophosphoric acid at crystal surface also block the further growth. Now the most important thing is transport of mineral ions to the mineralization front. Basically, the transport of mineral ions take place to intercellular transport. Tissue fluid is supersaturated in these ions, so fluid simply needs to percolate between the cells to reach organic matrix. Mineralization transport of mineral ions more likely to occur between osteoblast and odontoblast. In case of enamel, secretory said ameloblasts restrict the passage of a calcium, so majority of the calcium entry occur through the transcellular route. Sequestration of calcium to Golgi apparatus, mitochondria, and endoplasmic reticulum is a safety device to control calcium concentration of a cytosol. The possibility of transcellular transport is detected by the particular circumstance the cytosolic free calcium ion concentration cannot exceed 10 power minus 6 mol per liter because the greater concentration would cause calcium to inhibit critical cellular function leading to the cell death.